Okay, welcome back for the final projects for bioinformatics. And in this lecture, I'm going to go over all of the deliverables, sort of the schedule, where to get your data, what you'll need to do in order to finish the final projects. So let me go ahead and share my screen. And I'm going to start out in Canvas, just like you probably are. This is the videos and media section for module 8.2, and you're watching this instructions and deliverables video. You'll want to go ahead and download this instructions and deliverables PowerPoint, and that's what I'm going to switch over to for the rest of the time. We'll go back and forth as we move to different websites and things, but just to give you an overview, whoops, let's swap display. There we go. These are the deliverables in a nutshell. So an Excel file is going to show the sequence IDs and links for entire genome assembly. Then the results files from your Earl Grey runs are going to go into a directory on this site. A lot of this obviously isn't going to make sense right away, but this is what we're going to walk through. How do you get these deliverables? So one person in the group will make a directory in this shared location on bridges. And then every person in the group is going to upload their or move their files from their local directory into this location. They'll compress them all and then move them over, just so everyone has access to the summary files for your group. Okay. This deliverable number 2 is going to be due by December 3rd. That way, everybody has access to the results in time to do some of the summary stuff, just to make sure everybody's motivated to not wait to the last minute to get their initial analyses done, because there is some work to summarize afterwards. Third deliverable is a PowerPoint file. There's a template on Canvas for you to download that. It's really, it could be as short as 3 slides. We'll look at that in a minute. And then the last thing is the research summary. That's what will probably take the most thought and time as you answer all the questions that are in that Word document. Okay, that's also posted on Canvas for you to download, and we'll look at that in a minute. Okay, so the instructions. First thing you're going to do is go to your group's homepage. So I'm going to switch back over here to Canvas. And in Canvas, I'm actually not totally sure how you see this as a student. If I go into student view mode, that doesn't work. Yeah, so if I go in, I'm in student view, and I think I go to people, should be a groups tab. And then you will find your name, and maybe it only gives you access to your group. This is the thing, I can't exactly see it how you see it, because I'm not a student in one of these groups. Okay, so I'm going to leave the student view just so I can show you. If I go into one of these, and depending on which section you're in, these groups will be named differently. In the online section, for example, these are just group 1 through 20. Okay, but if you go into the group, I'll click on the thing, and I think you may automatically go to the home page. Anyway, the group's homepage is kind of blank looking, but you'll need to go to pages. So if you go to pages, and this is described right in the PowerPoint, this is what I mean by navigating to your group's pages. So that page, and actually I'm going to switch over to this one because this will line up better with what's in the slides. The Dendroctonus frontalis genome, this is southern pine beetle. So I'm going to go to that page, and what you'll see, just like on the slide, is information about my species, this is the name of the genome assembly, and then there's a link to take you to the website where you can download that. It also tells you how many chromosomes, and that'll be important in a minute. Okay, so right, that's what I'm showing you here. We're going to use that link to go get the genome. Okay, so that takes you to NCBI's genome page for this thing. Not all of the groups, some of the groups that may be a direct download just because some of these are from faculty members here that have recently done a new genome, and so we're going to analyze it for them. So that link will take you to the location where I want you to get the data, or it may just automatically start downloading it for you. It's fine. I put the links there so that we could be sure that everybody was working on the genome that I wanted them to. So if you're at an NCBI site, you're going to click download. We're going to get the genome sequences in FASTA format and leave that on all. You can leave this file name or change it, whatever you want, but it's going to then go ahead and download a zip file. And for me, you can look over here, and it downloaded this NCBI data set. You can see. And I just double clicked that and it unpacked it into a folder. Double click again, double click again, double click again. They really bury this thing deep in a directory structure. But I double click one more time and I'm finally at this genomic.fna file. That is my genome. And you can sort of tell as you were going, if you go through those, this is the one that looks like it's a genome file size that's something like you would expect for a whole genome. So 175 megabytes. This is actually a little bit of a small one, but I tried to give small ones to everyone, small-ish, just so that we could be sure that people's analyses run in a realistic amount of time. Okay, so I've got this just to make my life easy for some downstream steps. I'm going to take it out of that whole directory structure and just drop it straight into my downloads folder. So now it's not buried in those NCBI subdirectories. I don't need those anymore. I'm just going to trash them. So that's my genome. It's on my local machine. Whoops, wrong window. Switch back over here. Okay, so that's step one. Step two is to parse the genome for analysis. So even these new good genomes that I'm assigning you, their chromosome scale, I picked them based on that filter that we have to have chromosome scales assemblies, but that doesn't mean that all of the scaffolds in the assembly are attached to the chromosomes. So we're going to limit our analyses to the things that are really the biggest scaffolds and have the number of chromosomes we expect based on cytological data or whatever. So back on that Canvas page for your genomes, it will tell you how many chromosomes we expect that organism to have. So in the case, the example that I'm showing, southern pine beetle has 8 chromosomes. Okay, so we're going to combine that information with the length information that we're going to look at in a second to pull out just the chromosomes from this genome. Okay, so how do we get those chromosomes? Well, this is where you've got to go back and rely on what we did earlier in the semester with BioPython and how do you parse a FASTA file. So you can do this on your local machine or on Bridges 2. And in fact, really only one person in the group would have to do this because once you parse this and split individual chromosome files, it doesn't matter who does that. You don't all have to do it individually. You just have to decide who's going to get which chromosome. But whether you're all doing it and pulling your own individual chromosome, or one person is doing it and splitting all the chromosomes and then sending them to people, that doesn't matter. You can either do that on your local machine, or you can do it on Bridges 2. If you do it on Bridges 2, you'll have to go through the steps to install BioPython on Bridges 2. So I've given a link. This little graphic for BioPython is also a link that will tell you, take you to the site, and there are instructions there about installing BioPython. One of the goals of the final project is to see how much you have absorbed over the semester, and also sort of prove to yourselves how much you've absorbed over the semester. I think this should be easily within your grasp, given what you should know at the end of bioinformatics, to go onto a remote machine and install some new software. Okay, so doesn't matter if you're doing it on Bridges 2 or the local machine. Ultimately, what we want to do is create one FASTA file for each chromosome. So let's go ahead. I'm going to switch over. I'm going to actually show this to you using if I can find my window. Yeah, here it is. I'm going to actually show this to you using a Jupyter notebook, but you can do this from the terminal just as well. So this looks very much like something we did earlier in the semester, where we were using BioPython to import and parse a FASTA file. So I've written some code here. I'm not going to actually go over that to explain it. I expect you to be able to understand it at this point, and or write new code to do it all by yourself. In this case, right, I need to have an input file. And in my case, right, that's my genome. All I'm doing here is copying the file name, and I'm going to paste that in here because that's my input file. Right. And I should also say this Jupyter notebook is in my downloads folder as well, so I don't have to specify the path there. It's enough for me to just tell it the file name, and I could simplify the file name, but whatever, copy paste works just fine. And so what this is going to do is use the bioseq.io module to read this as a FASTA file, and then first of all, I'm going to have it print every record ID and the length of that record. Okay. And it's probably not clear why I need to do this yet, but let me switch over here just to tell you what we're looking for, because deliverable 1 is the Excel file showing the sequence IDs and the length of the entire genome assembly. Okay, so that's what I'm going to generate right now. And also, just for context, right, this is the first slide in the PowerPoint. This probably didn't make sense at the time, but this is the first deliverable. We need to have a file, and you're going to submit this in Canvas, an Excel file that has the sequence IDs and the length for the entire genome, the lengths of each one in the whole assembly. So this is what we're trying to get to. And so my code, keep losing my windows here in the stacks. My code is going to first loop through that genome and print every record ID and then the length of that record. So if I run this, you can see these are the sequence IDs in that FASTA file, and then that's the length of the sequence for that ID. So what I'm going to do is copy all of those rows, and you'll see why in a minute. I'm going to copy them. I'm going to stick them in a new, go away. I'm going to stick them in a new, whoops. So there's a little bit of a challenge if you try to directly paste into Excel. So I first, little cheat code here, I'm gonna paste it into a text document. So you can see I've got all my things that I just generated in Python that parsed the FASTA file. Now, if I copy from that text file into Excel, now it gives me actually this little import wizard that is going to let me split those columns. Okay, this is just a shortcut cheat code that I learned. There are many ways you could accomplish this. This just because I've done it so often, it's fastest for me. Okay, so now this is what I needed. I've got all my sequence IDs and then the length of that sequence. Now, depending on the genome, these aren't ordered perfectly. There's a pretty good chance because remember our genome that we're supposed to be looking at, Dendroctonus frontalis, has 8 chromosomes. So you can see the first 8 here are gigantic relative to the rest, right? So I could even put commas there just to make it. So you can see these are, the biggest chromosome is 42 million base pairs. The smallest chromosome is 12 million. And then there are lots of other scaffolds here that are, 15, 16,000 or less. There's one big one here. But that when I see these big ones, it also makes me a little nervous that this isn't in perfect order of size, right? Well, it's clearly not in perfect order of size. So the deliverable asks you, okay, to have it sorted by length with the longest one at the top. Once I've got it in Excel, that is super easy, right? I just highlight that column, expand this a little bit so it shows up. I'm going to sort, and I'm going to sort largest to smallest. It says expand the selection. Yes, I definitely need to leave that because I want to carry along the IDs corresponding to the lengths. So I do the sort, and you can see that the top ones, I think, all stayed the same, but down here, they're now from longest to smallest. Okay. Some of the genomes might have as clean a break here between what are chromosome scale and what are smaller unassembled scaffolds or unassigned scaffolds. We don't know which chromosome any of this stuff belongs to. So we are just going to keep these top 8. This file I would save, and then that's going to be one of the uploads. That is the first deliverable right there. I just generated the first deliverable. Okay, so the next thing, and this is not an immediate deliverable, but you need this in order to proceed with the Earl Grey transposable element annotations, is to get each of these chromosomes into its own FASTA file. So go back here to my Python notebook, and again, you don't have to do this in a notebook. It'll work just fine from the terminal. It just makes it easier for me to show you what's going on here. You can see I had some stuff commented out earlier. Now what I'm going to say is if the record, and let's just pull this first one as an example. Let me get the name of the first chromosome. And this is the name of the chromosome. And then at first I'm just going to let it print the same information, but it should only give me the info for that one chromosome. And in fact, yes, that's what it did. Okay. So now instead of printing that to the screen, I'm going to have this code add that whole chromosome record. I initiated a list called chromosome record, and now I'm just going to append this FASTA record because I know it's only seeing or only reporting this record. So now I'm going to write that to this thing called chromosome record. And then this next cell, oh, better run that. This next cell will open a file called output file and write using the seek.io from BioPython that chromosome record in FASTA format. So if let's actually call this output file.fasta. And in fact, don't call it that. Let's call it after our chromosome. Okay, so, right. So now we should get an output file named after the chromosome in FASTA format when I run that cell, and you could see it move over here. And so indeed, it wrote that file. So if I wanted to look at it, I could let's look at that in my terminal. And that's in my downloads folder. Let me blow this up a little. So I'll cd to downloads, and then let's just look at the top part of it because it's pretty big. Let's do head on CM. Okay, and so here, that's the top of that file, and it's exactly what I want. It's got my FASTA header and all the sequence for it. So I'm ready to go with chromosome 1. That's my data that's going to be input into Earl Grey to do the TE annotation on that chromosome. Okay. All right. So we've got deliverable 1 done, and we've got our, at least a pathway to get each one of the chromosomes. And I guess also note here, I was showing you, if you scroll down and look at how many lines are in that Excel file we just generated, there are 373. That is the scaffold number. Like, that's how many total scaffolds, including the 8 chromosomes, are in this genome. Okay, so we've done step 1, we've done step 2, we've parsed. Now the next step is to run Earl Grey on each chromosome. Every chromosome has to be completed by exactly 1 time. Each member of the group has to analyze at least 1 chromosome. So most of the groups are 6. There are a few groups with 7 members. Many of the genomes have 10, 11 chromosomes. There's one genome that I think has 20. There aren't very many that are big, but don't feel like you're getting cheated. It's not that much extra work just to add one additional chromosome. So all that is to say that some members of the group will have to do multiple chromosomes. Just don't do any of the chromosomes more than once. The reason all this stuff is in red, the reason that's important, that we're not repeating the analysis of any of the other students. We're not sort of I don't trust the analysis from somebody else, so I want to do it myself. We don't have enough compute time to do that. So transposable element annotation is really computationally intensive. And so we are probably going to burn through 100,000 compute hours all total. Now, that might sound scary, but because we're running 128 cores at a time, everything you do is multiplied by 128. And I expect that most of you, since we've broken it down into individual chromosomes for analysis, it's probably going to run in an hour. And some of the really long chromosomes, when you get into 100, 200 mega base chromosomes, those might take 5, 6, maybe 10 hours to run. Okay. But all that is to say, any of this red stuff, it's important that you don't duplicate runs. So I can see who's running what in the allocation. And if I find violations of this, individuals' grades will be dinged for that. Okay, because we run the risk of running out of time, and then the whole project is going to be in trouble. Okay. Uh, since you're not allowed to be duplicating effort, that means if somebody in your group is not completing the Earl Grey component here, so they're not getting their stuff done by December 3rd, they're going to be individually penalized, right? If they don't get their summary data into that directory, which is the next deliverable, then you just move on without them, and you'll have to do the analyses without consideration of that chromosome that didn't get done in time. Okay, so that gets me to the next deliverable. I put this in purple, trying to make this as noticeable as possible. Deliverable 2 is to get your summary files from the Earl Grey run into this directory on oceans, okay? And I'm going to show you how to run Earl Grey next. You miss the deadline, be a 20% reduction in your individual grade. If you're not clear, don't remember how to create a compressed file for your summary files, right? That's in Beginning Bioinformatics Part 2, going back almost to the very beginning of the semester. You hopefully, the end of a bioinformatics course, this is something that you can just do.